all right, New Zealand accent. So why? How? These are the questions of how do we regenerate soil. The curiosity and the wonder requires asking these questions. One day I was driving with my son. He was 4 years old. I was taking him to kindergarten. And the scene looked a little similar to this. We were living on an avocado orchard in the Bay of Plenty. And as we traveled to school or to kindergarten, he turned to me and he asked, "What is that white stuff coming out of the mountains?" Now, I'm a horrible parent. I was ready, right? Well, it's the inversion layer with transpiration coming out of the stomata. And I stopped and I said to him, "I wonder." And what he said was it was dragon smoke. And I'm like, oh, that is so much better than my answer. So much better. And I think we've been programmed to look for these answers when what we really need to be programming ourselves in is how do we ask better questions. So this operation here is Riversun Nurseries in New Zealand. They grow about 5 million rootstock for viticulture, kiwi fruit, avocados as well. They're a premier supplier. They are the largest one in New Zealand. And what you see here is a standard practice, all right? So they will make mounds and furrows. They then lay it with black plastic, right? That makes the soil nice and warm. Then the rain can't get through, so you put irrigation tape underneath the black plastic. Then during summer, it gets too hot. Uh-oh, paint it white, they do. Excellent. So we had a conversation about why. Why is it you're painting it white? Well, it's too hot. Why do we need the irrigation tape? Well, it's too dry. Why are we doing this at all? Well, we need to control the weeds. So what we trialed instead is we rolled bales, small bales of barley straw down the middle of these rows. And what we found was we got the same weed control. Actually, not in the first year. So the first year we rolled it out and all the barley straw sprouted. Not what we were thinking. So the second year we actually sterilized it, steam sterilized it, did a beautiful job controlling weeds, and they didn't need the irrigation tape. They didn't have all this waste to dispose of, and it saved them hundreds of thousands of dollars. We did the same thing with their fertilizer pellets. So they have quite a large tablet that they will put below every single rootstock plant, and they had been doing that for 30 years. It was costing them about $180,000 a year just in the tablets. So I said, why? Well, we've always done it. Oh, right. Should we try that again? Let's see what happens if we don't put the fertilizer tablet underneath. And you know what happened? No difference. No difference at all. Wow, wow. All right, so we stopped doing that. Very wise, very wise. So you'll be seeing what's happening in Australia at the moment. It's actually really exciting for me to see that Americans realize there's this place called the rest of the world. Sorry. All right. So I was traveling through Western Australia early this last year, so February, and I took a train, or I drove actually. I took a train from Townsville, which is right up here, down to the Sunshine Coast. It was about an 8-hour train ride. I then drove from the Grampians, which are about here, then inland to a lot of the cropping areas, then to Melbourne. I drove along this coast until I came up to Canberra. The entire time, there's no ground cover, right? That soil was so desiccated, so dry. The cattle and the sheep looked like skeletons that someone had stretched leather across. And by the time I got to Canberra, it was so emotional. Like I really just was like, we are so stuffed, right? And then I got to a property called Jillamatong. This is Jillamatong. They're about an hour and a half from Canberra. What he's practicing is keyline design using a yeoman's plow, contour lines. So they're actually running contours to make sure that when someone says, how much rain did you get? What do you want to be able to say? All of it. Not 6 inches, 4 inches. You want to be able to say all of it, and it functions like a sponge. He can say all of it. So what he's doing is a practice called natural sequence farming, which is promoted by Peter Andrews. He wrote a book called Back from the Brink. He actually wrote 2, looking at how do we rehydrate these landscapes. I'm just going to move this. Right, they were putting compost into the contour lines onto the contours, which was really interesting. So when they got a bit of rainfall, I'm going to break something. When they get a little bit of rainfall, it actually moves the compost down through their system. They did something called a leaky weir. So if you see in this image, what they had were these massive eroding gullies. So they put in these weirs designed to slow water down. And so they filled these gullies with water. And then he's using adaptive grazing. So this was last February, after being in an environment that there was no grass. It's just dust, right? It's just dust unless people are practicing regenerative practices. From his property right now, so he burnt, and he burnt twice. So he burnt in December. The helicopters could actually come in. They had water. They could take water out of the weir. But the local council's actually buying the water from him because the rivers and the dams have all dried up. There's no water at all. This guy's producing 5,000 gallons of water as a river which runs out of his property that starts on his property. How do we start to create these systems that are hydrated, that absorb when you get that rainfall so that you've got rain, you've got moisture coming into dry summers? What we're not seeing flash flood drought like it's a natural occurrence. It's a land management problem, right? How do we start to do this on whole catchments? So they actually got the Canberra government very excited about this, and they have funded a 23-mile-long catchment restoration using these practices. And if they work, which of course they're going to work, they're looking at rolling it out nationwide, which is very exciting. As we drove around his place, we counted 12 different mushrooms. And I was so excited to see all of these in the middle of that dry. This mushroom here, it's called Cordyceps gunnii. Now, some of you may be familiar with cordyceps. You might be taking that as a pharmaceutical. Some of you might remember in the 2000 Olympics, the Chinese women's running team were banned for taking this particular mushroom. It gives you endurance and stamina. There's something else that the Chinese think it gives you. What's the word? Libido, that's the word. Thank you. All right, so they've actually been making a tincture out of it and giving it for free to all the guys in the local town. They have been banned from taking it anymore. Apparently it's very effective, all right? But mushrooms like this could actually be a stacked enterprise. He's collecting about $180,000 worth of fungi, these cordyceps mushrooms, off his own property. Right? What they are is they're entomopathogenic fungi. Can we say entomopathogenic? Entomopathogenic? Some of you know. Okay, right. So ento means insect, pathogen, which I always thought was like disease, but pathogen literally means to suffer. So it's insect suffering fungi. You guys have them here in Iowa. I know where I am. No, I know where I am. I do. Sometimes I don't, but I do. I know where I am. All right. So this one here is actually growing inside a fly. This is Cordyceps variabilis. The Bovaria bassiana, or white muscadine disease, you'll see it turning up like a white fuzz. And metarhizium turns up as green muscadine disease. You can buy these as commercial products. We are using them in the transition from high input systems to regenerative systems. We kickstart it where there are insect pests. And many of you have so many insects chewing away underneath the ground that you're probably not even accounting for, all right? They use metarhizium to spray on bees to control varroa mite, all right? So it's a very selective fungus. Super cool. So this photo here is actually from Kim Deans, who's one of my consultants in Australia. And what you're looking at is a fence line photograph. What you're seeing in Australia at the moment are these huge clouds of dust, right? Big dust storms. That is not natural. When we get biological activity happening in soils and you get all their glues and mucus and vomit and spit and poos and wheeze and all that stuff, soils stick together. So what you see is her property, which is in the foreground, is not blowing. She has no grass because she burnt out in February as well, or January, right? So they lost all their grass, and the frustrating thing was, as a regenerative producer, you have more grass. So you're the one that's going to burn worse in these fires, and they burnt black. But they're the first ones to come away, and their soil is not leaving. consider that the very fine stuff that you see moving and blowing, that's your humus. That's your organic matter. That blows before your clay or your silt, all right? When you see people out cultivating and a bit of stuff is moving, that's your gold blowing away. Now, they got an inch of rain, very excited. Did the neighbor get an inch of rain? No. What you're looking at is a lake at the neighbor's place, all soaked in on their property, right? Same elevation. It's all about land management. Can we create that sponge? Can we create that stick? So one way I often think about soil is actually how do we relate this to our human body? So when we eat food, we're going to chew it, make it smaller. We coat it in saliva and enzymes. In soil, that'll be your earthworms, your termites, your ants. They're chewing, making smaller. We pass that through to the stomach, acids in there. Same in the soil, you've got your fungi and bacteria making some of the most powerful extractant acids. And then we pass through that gastrointestinal system. In there you have 3 pounds of beneficial microbiology. Well, hopefully beneficial, all right? Some of you not so much, all right? So we have all these microbes. Their job is to break that food down, to make it even smaller so it can then pass into the bloodstream. In that process, though, they're releasing things like serotonin. 90% of the serotonin in your body is made in the gut by gut microbiology. So how you feel in the morning is actually determined by the types of microbes that you have. And what we're starting to discover is how little we know. And it's the equivalent of having a flashlight and we're standing on the edge of a void and we're shining it. And what we can focus on is what we're looking at in terms of research. But there's so much that we don't understand about what's happening underneath soil, which is why I find it so incredibly exciting. But we can relate this all to what's happening with our bodies, right? So they make us grow, regulate health, provide vitamins, enzymes, hormones. They help your whole body to function. And what they find is that disruption of gut flora is related to over 80%, I love the applause, it's just fabulous, to over 80% of disorders that we're facing and 100% of immunological disease. So anything to do with inflammation, rheumatoid arthritis, gut problems obviously, dementia is all now being related to what's happening with your gut. So here's a small list of some of the things that relate to gut microbiology, right? From acne to asthma to cancers, cavities. Now looking at their list, is there anyone in this room that's never had any of that? Sometimes there is one person and they're a liar. No, I'm sorry. No. I'm sure there is. It's fine, all right. But what we're seeing, and this is what I love coming to the States, is you have this whole aisle and half of it's dedicated to things to make you poop and half of it to stop you pooping. I didn't even know that was a thing. In New Zealand we have like 2 products, all right? That human gut microbiome in America is significantly broken. All right, what kind of things have we done to break it? Sugar, so a narrow food source, antibiotics, processed foods, preservatives, pesticides, absolutely. Your addiction to pharmaceuticals, holy Toledo. All right, all of these things, the way you were born, were you raised around animals? Did you have a cat and a dog? Did you go out on a farm? Did your parents let you play in the dirt? All right, or do they make you stay inside and be all sterilized? All right, we are influencing that gut microbiome all the time. Who breathes on you? The very first person can inoculate you with either bad strep throat or good strep throat. So I was studying soils in 1999, and one of my lecturers had made a tablet called Bliss. And you will take this when the baby's about to be born and everyone chews it, and then when you breathe on that baby, you've inoculated them with the good strep organisms for their whole life. So cool, all right? But you set people up for life, and then it's really hard to then change some of that. Guys, not eating diverse foods, holy Toledo. For me, here, trying to eat food, a little bit of iceberg lettuce is not green stuff, people. All right? It's not. It's not. All right, you want at least half of your plate full of green stuff and not just iceberg lettuce. All right? There's about 14,000 edible plants that we could eat, and commercially, you guys are eating less than 30, all right? There used to be about 300 different varieties of lettuce, and I don't know what it is now, but it's like 24 or something, all right? We're losing diversity in all of the foods that we eat, so you can have the pretty iceberg lettuce and the, whatever that big red tomato thing is. Ugh. Anyway. So, we're blowing this microbial bridge, right? We're blowing that bridge in our own gut, and there's consequences with all these things that are called modern society. And we're doing the same thing to our soils. So our soils have indigestion, constipation, gas, dehydration, and if you go to New Zealand, rampant diarrhea. All right, awesome. Because that whole gut function has broken down. So you guys lose about 6.9 billion tons of topsoil per year. It is your biggest export. Woo! All right. And it's moving a lot of the time through the soil. Like you're not even seeing it. It's happening every day underneath our feet, right? And it's uncalculated for. You lose on average about 4 to 5 tons of topsoil per acre per year. But I'm hearing stories of some of these properties are losing over 50 tons per acre per year. And you see it when those heavy rainfall events happen. Right? We need to restore that microbial bridge. Now, microbes are everywhere. Even if, like, you were just to disappear and just leave behind your microbes, we will see an outline of your bodies. Right? Bacteria, nematodes, fungi, some of you. Fungus, maybe in the room. All right, so we've got this microbiology that coats us, and there's a reason for that. Right, and same in the soil. So there's a huge number of microbiology in soil. So say we took a teaspoon of soil from a disturbed property, like a teaspoon. You're going to see maybe 5,000 species, which is the equivalent of maybe a Jersey steer per hectare. All right, so you cut that in 2.5 to get that into an acre. Not very functional, that Jersey steer at that point. Anyway. But most of that diversity are disease organisms. Whereas we start to bring life back into these systems, regenerate, bring carbon, diversity, then we can see as much as 25,000 species in that teaspoon. Species, not individuals, right? Talking about millions, billions. But the biomass is the equivalent of about 5 Angus bulls to 10 Angus bulls per hectare. Do you want the Jersey steer or the Angus bulls? Jersey steers are quite yummy, I shouldn't say that. Anyway, so there are tens of thousands of signaling molecules that are in soil. And the reason for this is, imagine, you're a teeny tiny little bacteria, like this might be the surface of your cell. You're totally blind, and everybody's out to get you. So you're like signaling, all right, are you my friend? Are you food? All right, are you going to eat me? And these receptors tell them what's happening in that environment, what's happening nutritionally, temperature-wise, moisture. But they're signaling using things like proteins and hormones, different types of elicitors, enzymes, and the plant defense molecules, salicylic, jasmonic, chitinase, and proteinase. All right, these signals are happening all the time, and they can actually alter how that cell expresses itself using a process called epigenetics, right? So think about every single one of your cells has 100,000 receptors on the surface waiting for a signal. So in soil, they're communicating all the time. So you think about these cordyceps mushrooms, like they're going to blast out of your bodies. There are these microbes happening in soils all the time. They're communicating to each other to get away, find food. Some of them pretend, like they hide and they pretend to be other types of bacteria. They actually, recently they had this video that came out, and it was protozoa, who are also blind, hunting a nematode down like a pack of wolves. They're doing it all with electrical signals and biological signals to communicate, right? No eyes. So the process, one of the processes that we look at in terms of communication is called quorum sensing. Right? So we've known about quorum sensing for a long time, since about the 1950s. They were looking at bioluminescence of plankton. You know, like suddenly you splash in the water and everything lights up. How do they signal to each other that it'll light up? Right? And it's these chemical signals, these quorum signals. So they discovered the same things happening with ants and with bees. And now they've discovered, probably about 15 years ago now, that at any one time, let's say you have strep throat, but you're not virulent, and then at some point suddenly you've got a sore throat, right? You've got an infection. It's a chemical signal that switches that bacteria on to make you infected. So you can imagine the medical world's very excited about this. So they discovered, for instance, and it was about 10 years ago, you can drink a drink with the parts per trillion of a chemical signal for cholera. You can drink it. It tells the cholera that there's too many other cholera here, and they flush out of the host. Now, I don't know why, but they're not making it freely available. Go figure. Right, but it's parts per trillion. Very, very, very small amounts that are turning on biology. And we are using this on rangeland here and in Australia to turn microbes on. So this quorum sensing process includes some of the aromatics, exudates, hormones, pheromones, all of this stuff, right? That there is a plant root. Out the tip, it's sending out sugars. It's sending out biology actually in that. It's sending out fatty acids and these signaling proteins to say to a microbe, hey, I really need this kind of mineral, or hey, I really need this kind of defense molecule, and the biology respond. So just like it is with our human body, over 80% of plant health and nutrition is driven by this microbial bridge. When I went and studied soil science, there's a manual, and it's still the same book that they use today. There are 4 pages on microbiology in there, and that's what people are learning when they learn about agronomy. They don't learn about the microbes, or they might, the diseases, but we're not learning about how do we restore the microbiology in the system. And what we find is the more diversity you have in there, the more signals that start to happen, and the more gene expression, which is resilience. So we do that by thinking, all right, I'm going to put diverse crops out there. Maybe I'm going to have some cattle. Maybe I have sheep or geese, whatever. Like getting diversity into that system, the system responds. And one of these interesting elements of the system responding, if you imagine that this plant here on your left-hand side, this is a plant that has a broken microbial bridge, right? It's communicating because it's got something like botrytis or sclerotinia or amylaria or whatever you got here, take all. And it's communicating, but there's no one there to respond, and it ends up with a disease infection. Diseases are present all the time, right? It's not about whether or not you have a disease, it's about do you have the beneficials to suppress it. So on the right-hand side, that plant signals to the biology, which in this case is trichoderma. So trichoderma is a fungus that eats fungi, and we're using this in our programs as well. And it will signal to that trichoderma, and the trichoderma responds with a chemical signal that enables that plant to induce a systemic response. That the plant can now produce salicylic acid or ethylene or jasmonic acids. These are the hormones that plants use for plant defense. They can't do it by themselves. So it's like the plant's stomach is outside of its body and its immune system is outside of its body. Right? It can't have everything here. I'm just a little plant. I'm going to ask my community around me to support me, right, and make me healthy. So one thing that we really want to see in our regenerative systems, are we building soil? Are we building carbon? Are we building resilience? Is these structures. The rhizosheath, or what we call the Rastafarian rootsman, right? Have we got the dreadlocks, right? If you don't have a dreadlock structure, then you're missing all of these benefits. Because what it's providing is plant protection, nutrient uptake, growth, it's feeding your microbiology, it's building carbon, building resilience, and it's a buffer against things like sodium or against changes in pH. So your pH might be 9, but what that plant actually experiences is 7. All right? So in here, in that sheath, it's a different pH. It's all microbially remediated. You might have a pH of 5, but the plant's experiencing 7. In Australia, they've got really big issues with, I'm going to practice this in my head, aluminum. All right, so aluminum's a big issue, and actually a lot of their grains are full of aluminum. The reason for that is when you have a look at these rhizosheaths, they don't have any, right? They have clean, white roots. If you have clean, white roots, now go home. No, this would be horrible right now to dig a hole, wouldn't it? All right, don't dig a hole now. When your crops come up out of the ground, dig holes and have a look. Have I got a rhizosheath in there? Have I got defense? Because if you don't, you're basically hydroponically feeding that plant. You're going to need phosphorus. You're going to need nitrogen. The nitrogen cycle only works when we start getting these kind of rhizosheath development happening. Right? If you don't have that, clean roots, your system ain't working. Right? And now you're going to have to put some inputs in. So one thing we're doing is seed dressings. So seed dressings with different types of microbes or compost extracts or worm extracts. Right? It's easier to see these rhizosheaths on sand, but look, that plant's just germinated and look how big the rasta roots are. Right? You want to see that structure. The minute that that plant punches through, it's got that microbial protection. And a big part of this is this process which is called rhizophagy, that plant roots are actually exuding microbiology and they're feeding them, and then they'll actually grow over them and absorb their poor little friends' bodies. Horrible plants. All right? So like up to 30% of that early plant and its germination is all due to the fact that it's absorbing bacteria. Right? So it absorbs their bodies and takes up the nitrogen and the phosphorus and the sulfur and whatever it's liberated. But if you have a broken microbial system, you're now going to have to put down starter P. Right? You're going to have to put something down the drill instead of allowing the plants to access their own nutrients the way nature designed. So I just want to show this an example because some of the things I'm hearing about what you're dealing with around in Ohio, just kidding, is around soil structure and water holding capacity. Right? So these two orchards, they're side by side, they're separated only by a headland, same soil type. This here is best practice, right? So it was a research center, plant and food. They're doing all the things you should do to be a good farmer, right? You're going to make it pretty with a bit of glyphosate. You're going to irrigate because now you have to, right? They're not using compost because that would be obviously quite insane. Whereas this guy, and they took photos of crazy trees on purpose. I never saw these crazy trees while I was there, but anyway. So he's using compost, he's allowing pasture to grow, he's not irrigating. He has 4.4 pounds of carbon per yard, which translates to 11,000 tons more carbon in the top 10 centimeters, right? 11,000 tons more carbon just in that top bit, right? So it's under where the compost is being applied. He's growing carbon. Now what was really cool is they used the machine to measure the pore spaces in the soil. So what you're looking at here, the white is the empty spaces and the dark is the soil, right? And we're looking at about 4-inch depth, okay? So you notice this is the IPM best management. See, there's hardly any pore spaces, it's not very connected. This one here, 80% of it is all connected. Now, what do you think happens when you get a heavy rainfall on this? Where does the rain go? Yeah, it's going to run off, right? It's just going to go straight into the drains. What about the water in here? You're going to put, I don't know how to do American. I'm going to say a litre, all right? So, a litre. You're going to put a litre of water on the top. Where does it go? Just soaks in, right? And so when they measured it, 2/3 of that, or a third of that water was held on, right? In that 4 inches. The rest of it came out the bottom, but a third of it was held on. So then they did what scientists do best, is they were like, let's do 2,4-D. Right? What happens to 2,4-D? Yay! All right. So they put 2,4-D on with water. Where does it go here? It just goes right off, right? So in the Great Barrier Reef, this is causing massive problems because it's killing the reef, right? 2,4-D into the water. On the right-hand side, they put in the 2,4-D. They measured it. No 2,4-D. And in fact, the water came out cleaner than it had gone in. Right? So there was no chlorine, there was no fluoride, and there was no 2,4-D. So they went, all right, well, idiot forgot to do the 2,4-D, do it again. Did it again, no 2,4-D. Where's it gone? They ate it, right? They bioaccumulated or bioremediated that 2,4-D, and even when they broke it apart and measured that soil, they couldn't find it, right? So you can kind of think like there's Wiley the worm, and he's eaten it, it's in his body, and then he dies, and his sister eats him, and they just keep it out of circulation. Right? The problem can be is people are taking soils like this and turning them into that, and we're seeing heavy metal release, pesticide release. So seeing properties that might be organic 4 years after intensive chemical use, and those chemicals are starting to come through into the plants. Right? You've got to develop a rhizosheath to defend yourself against those kind of chemicals. Right? So the total connected area in that conventional IPM is this. burrow. It is not a worm. Who's made that burrow is going to be like an army cutworm. It's going to be a cricket or a cicada, or I don't know what you guys have out here. It's my first time. What do you have? What kind of bugs? Grasshoppers? All right. So most of those insects spend most of their lives under the ground, all right? And it might be 8 years before they even hatch, or 13 years before they hatch. In these soils, those entomopathogenic fungi, your metarhizium, your Bovaria, don't exist, right? They will not survive in compacted soils, but these guys will. You're creating the conditions for insect pests when we start to compact our soils. Whereas the organic orchard, that whole thing's connected. There's no limitation to nutrients or water movement or root movement, right? Those roots can go wherever they want to go. So one of the effects that has is on temperature. Start measuring your temperature, and I call this the smug test, all right? I'm going to measure my temperature and then the neighbor's loses, all right? So typically what I find is our place was significantly warmer, like 30 degrees warmer at the start of spring and 30 degrees cooler in the middle of summer. What does that mean? What does that mean for you? Less stress, all right? You'll see that the temperature in there is buffered, like being in a big old straw bale house or whatever, right? The temperatures are buffered, and this is the most extreme example I've seen on it. Regenerative on the right, NPK on the left. All they were using on the right-hand side is worm extracts. The temperature here at 1/5 of an inch is 168 degrees Fahrenheit. You couldn't even you couldn't rest your knee on that soil to take that temperature. How stressed are those plants? And you can see how stressed they are. Now, on the right, 90 degrees. It's still blinking warm, right? But they'd had 3 days over 120 degrees, right? Western Australia. It's biology that buffer or give you resilience against temperature, right? And we will see snow melting on these fields. We will see frost not settling because of this microbial effect. Has anyone ever felt like that? Any day, all right? So part of my process is looking for what puts a drag on your system. What is slowing down your soil health goals, right? And part of our triage system and part of our thinking around what is putting a drag on the system is we look at the 5 M's. So your minerals, what's happening with minerals? Microbiology, your organic matter, your management, and your mindset. And if you're not sure if it's you, it probably is, right? So a lot of the time what we're dealing with is I feel like I'm a bit of a psychologist. We spend most of our time dealing with what is it that stops you from taking action. Why is it that you're muddling about here and muddling about there and not actually committing to something? What is your thinking processes? But actually, in a lot of environments, we're finding it's the microbiological system that's being shut down. So one of the organisms we talk about in soil, or two of them actually, bacteria and archaea. They're actually totally different organisms. So archaea are not related to bacteria at all. They look the same. Archaea means the ancient ones. These are the first organisms that landed on the planet, and your great-great-great-great-great-great-grandma, basically, all right? So they're the oldest organisms. They're involved in disease suppression, holding on to nutrients. So I think of these guys as the fertilizer bags in the shed, unopened. Not helpful, right? But they're good, nutrients, but not giving it up. They form the very small soil aggregates. So we start to see these soils that slake or fall apart, or you notice when you get a heavy rainfall how they just kind of go all smooth. That slaking effect is due to bacteria. Okay, so they're very important, but they create, this is one of the worst examples I've ever seen, very bacterial soils, also in Australia. But it can lead to compaction, right? We get surface crusting, you get soils that do not breathe, you get soils that do not hold on to water. They're also one of the key indicators for nitrates. So we'll see nitrate spikes. You get bust and boom populations of bacteria, and they release nitrates in that process. And you will see the nitrate weeds, pigweed, nightshade, nettles. I'm trying to think. I don't know. I've never been here. You guys have nitrate weeds, right? You'll notice these will be the weeds where you have been fertilizing or you've been putting too much manure on, or it'll be around your yards, right? Where you've got animal yards, these are your nitrate weeds. This bacterial dominance sends a signal to those types of plants to germinate. So when I go on a property, what I'm looking for is what kind of plants are growing, because they're telling me a whole lot about you and the previous owners and history, but it's our fingerprints are left by the types of species that are growing, right? So who makes it rain? Any idea? What? God. Very good. Right. So 40 to 100% of ice crystals contain microbiology, all right? There's a whole ecosystem up there in the sky. And one of these organisms is called Pseudomonas syringae, and what it does is it forms ice nucleuses, all right? That's that very first beginning process of forming snow and ice and hail, all right? But what they're important for is they form the frost on the leaf, all right? So you're seeing frost forming on leaves. It's a type of bacteria. So one of the things that we can do to reduce frost is we're going to reduce those free nitrates. All right, you've got nitrates in the system, there's more water in that system, that plant's ability to withstand frost drops. As we lift bricks, is anyone measuring bricks? Show of hands. Okay, you want to start measuring this stuff. Oh, there was like a whole section up here of like the awesome people. All right, so we're measuring plant photosynthesis. How well is that plant turning over? Because that turns into everything. Everything that leaves the farm gate starts as this process of sugars, which then turn into everything and build our soil carbon. Having biological activity on the leaves is going to help. This organism here is called Pseudomonas fluorescens. It eats Pseudomonas syringae, the frost-forming bacteria. So we're actually using these organisms. You can get them as packaged products, or there's another way where we can find them. So minus 6 degrees is what, 20? About 20 degrees Fahrenheit? Huh? Ish? I'm going to go with that. Okay, and we're using them in viticulture and horticulture and market gardens to protect against those out-of-season frosts or early or late frosts, right? And it might look like this, right? So we're getting that warming of the soil, you're getting microbiological protection on the leaf surfaces, we don't see frost, right? And these organisms, the Pseudomonas fluorescens, come out the butt of worms. The elixir of life comes out of the back end of a worm. Really, right? Those worms have remained unchanged for hundreds of millions of years because what they do is perfect, right? They send out plant growth hormones, enzymes, and all the microbiology that a plant needs to be healthy. It's like, I don't know, if there was some kind of magic plan, this is what plants need, and the worm's like, cool, poop. Anyway, and that's what they do. Right, now fungi, on the other hand, also important for disease suppression. They retain nutrients, they will give it up to the plant, they break stuff down. So I used to be a commercial composter, and I was making vermiculture on a large scale, and this is what my vermicast looks like. So vermicast is the worm poops. It looks like wood chips. Very high in fungi, very hard to find this stuff commercially, but we're using it as an extract. They form the large soil macro aggregates. So when we talk about aggregate stability, we talk about water holding capacity and all that air in the soil, it's driven by fungi. They're what holds soils together, and what they're discovering now, and Chico State University is doing this, Germany's been doing it, New Zealand's doing it, New Mexico State University is finding lifting fungal biomass and activity has more say on yield than any other factor. More than nitrogen, more than phosphorus, more than potassium. You want to play yield and profit, you want to start working with these guys. Oh, I can kind of touch on that. So one of the things that fungi actually do is they make crystals. They build rocks. They're building stable carbon in the inorganic carbon form deep down in the profile. So if we're interested around some of this stuff about getting carbon down and carbon drawdown, fungi are a big part of this picture. So this is calcium oxalate. So what we're looking at under an electron microscope are the hyphae of fungi, and what's coated in all of that is something called calcium oxalate. Right? It's a crystalline calcium in that's in a stable form that the plant then makes it, that the fungi then make that available to plants, right? And what we're seeing is a lot of our crops are very low in calcium. How do we know that? You're probably going to have diseases. You're probably going to have insect attack, right? As we start to live functional fungi, we see these things going away. As our fungi to bacteria ratio increases, so too does carbon. Fungi don't respire the same way as bacteria. You know, bacteria are kind of like zooming around and they're getting it on and respiring and breathing it off. Fungi are much slower, they're laying down chitin, they're building the system, and they respire far less. Right? As we start to lift those fungi, we will see less weeds. These guys require more of what we call the complex foods. So we're using humates, fish hydrolysates. We are using very finished compost that's got like wood chips in it. But think things that are hard to break down, your cellulose and lignin. And what we find in a lot of systems is we're either not putting this kind of stuff on, or those organisms that break down are not breaking down anymore. Right? So we're seeing stubble from 3 or 4 years ago. Right? What do we need to do to get these fungi active? All right, so I just want to share this case study. I'm not currently working with any cropping guys in the US, right? But these guys up in Canada, in Alberta, they're growing 35,000 acres of wheat, barley, canola, and peas. Average precipitation is 12 to 19 inches, but I've noticed that in a lot of areas, the average doesn't mean anything anymore, and it's been significantly lower than that. And in some areas, we're seeing significantly higher, right? So why would they come and change? Why would they come and talk to me? These guys are Hutterites. Why would they talk to me at all, right? So they were in the top 1% of producers in their region, all right? These guys are doing very well. But one thing they were noticing were things like market signals. So last year in the rural papers in Canada, there's full-page adverts put out by the canola board and lentils saying things like, we will no longer accept desiccated crops, right? That glyphosate is not a food additive. Did you know that? No, not for food. Anyway, so they put out a whole page saying, we don't think you should do that anymore. So they were seeing those kind of signals. They were having real issues with soil health. Their soils were becoming so compacted that it was costing more and more in fuel to get through, and then their soils are really wet. So these guys are at latitude 58. They're right on the edge of where it's possible to grow crops, right, before it just turns into all woodlands. Right, so grey wooded soils. They have about, they can have like 5-6% organic matter, so good organic matter levels, but it's not functional, right? It's not, the biology wasn't working, wasn't turning over. So this is what their soils look like, right? So 2 inches down, hard pans. See how the roots grow sideways? There's no mixing between these two different soils, and on these really sticky clays, right? So what they do is they get really, really waterlogged, and the snows would melt, and then really droughty and cracked later on in the season, right? So water became a real issue. AMF stands for mycorrhizal fungi, very high magnesium, sticky soils, low trace elements, low sodium.
Maybe what's happening is the radish is really packing them to the side.
No, maybe. You're making it worse.
Well, it's a good thing you built that shovel. Like weird clay right here.
The purpose of that is just to show you that like cover crops aren't necessarily going to solve your issues of tight soils like that. Like we took us ages to get that swede out of the ground. So what we did is this is our first year program in the wheat. So what we're doing is we actually work with a really cool humulite, the humate company, and they prilled this product for us. So we had 2 different prills. We're mixing them and we're putting trace elements in it. We're putting gypsum in it. What do you want gypsum for? Calcium? Sulfur? What does the sulfur do? Lots of things. So, amino acids, yeah, forms amino acids. In this case, very high magnesium, so we're using the gypsum to flocculate or move that magnesium molecule out and replace it with a calcium. So what it does is it starts to open up and fluff up that soil right below where the seed went. So we're dropping this prill, it reacts with a bit of moisture and it opens up that hard pan right below the seed. I don't care about influencing the whole thing. I want to give that plant the best start it can, because it's the plant that builds soil. Right? We're fooling ourselves that we build soil. It's the plant with its microbial relationship. So if we set that up for success so that it can punch down, oh, I won't do that. Oh no, I won't do that. So yeah, it was really cool to be able to put this together. We put sea minerals on, and a lot of people, when I'm dealing with sodium, are like, get out of town. You're not putting sodium on my property. Most of North American soils are deficient in sodium. And we talk about it, it's not a necessarily plant growth element, but it is for biology. It is for living cells. So just like we need salt and your cows need salt, so too do microbiology. It's an electrolyte. It gets soils very excited, all right? We see a growth response. Then we're doing seed treatment. So I've put mycorrhizal fungi in there and trichoderma, but the company lied, which we didn't find out till the following year when we reordered it, and they said, oh, we don't do that product. And I'm like, but we brought it off you last year. So not quite sure what we got in the end. And some phosphate solubilizing bacteria. All right, so helping to release that bound P. As a foliar, we put on 2.4 liters an acre, so a gallon is 4, so half a gallon, all right, of NPK, 10, 10, 10, fulvic acid. Everyone know fulvic? I saw someone at breakfast this morning had a big jug of it on the breakfast table. I'm like, you go, all right? So fulvic acid is a soft brown coal. It's very fine. It's just basically like an extract of carbon, right? So we put that in with our programs, and it feeds microbiology. This image here was taken pretty soon after that plant's punched through and starting to grow. We're starting to see that rhizosheath. Within 6 weeks, those plants were all down through that hard pan. 6 weeks, right? We did that because we're flocculating or opening up the soil beneath it. So you go, where's their nitrogen? Because it should say it on that picture, and it doesn't. All right, so they were using 110 units of actual N. In the first year, I dropped it down to 30 with no change in yield. You guys are still sitting there, that's good. No one's run out of the building screaming. All right, we were looking at their plant tissue test. Nitrogen was not their problem. If you have compacted soils, if you have low biological activity, that's what's causing the nitrogen problem. Right? High magnesium, tight soils, now we've got a nitrogen problem. Has nothing to do with actual N. Okay? So we dropped that nitrogen down. Their crop looked stunning, so they were really happy with how even the crops looked. Crop yield, things like peas. Oh, okay, I left that in there. I was going to take it out. All right, so this is one thing that we do, is we add fulvic acid in with our herbicides. Not residual herbicides like leaf-applied herbicides. We can drop that herbicide by 30 to 50%. Now you're off label. Funny thing was, that's what the labels used to be. Aren't now, are they? Right? So we can actually drop that rate. We add fulvic acid. It's a food for microorganisms. They sense that carbon's landing, and they open themselves up and go, oh, yummy, and then you kill them really, really effectively. Okay, so we are doing this across hundreds of thousands of acres. I hate saying we've never had, touch wood, never had a train wreck, all right? Find this to be incredibly effective, and it obviously cuts our costs, and we're feeding biology. So that's their pea crop. They were very happy with it. I don't spend a huge amount of time with cropping, but yeah, good, peas, yay. So this is the plant tissue tests, all right? So in the first year of starting with them, what you see is a whole lot of variability. So high sulfur, magnesium, calcium in the plant, very low copper, very low boron, and actually excess nitrogen. And what we find, look at the nitrogen, despite the fact we've gone from 110 to 30, right? Nitrogen wasn't their problem, compaction was. And when I see stuff about what's happening here in Iowa, I have to go second guess now. It's a compaction issue, all right? You've lost that beautiful volume of air. Your soils aren't acting like sponges anymore, right? They're turning all soggy and wet. That orchard photograph that I showed you earlier on, they can drive on those fields even after, say, 5 inches of rain, right? They have soils that bounce. And it's the weirdest feeling. Like you just go, whoa, these soils are bouncy. And then you walk to the neighbors and they're like concrete, right? How do we create those types of soils? So in the first year, we had a 60% reduction in spend. We're punching those roots through. The yield was about an equivalent, and we did this on 5 inches of rain. I've put failure, right? So failure, whatever failure means, all right? So the cover crops struggled because they got their crops in with a bit of snow melt and that kind of moisture, but they didn't get any other rain. So by the time they put the cover crops in, those seeds didn't get germination rains. Right, so we've got really patchy growth. They do about 1,000 acres a year, right? And patchy growth. So yeah, that sort of stuff can be expensive. So what we've done is we maintain yield, decreased costs, and we've increased water infiltration by 5 to 10 times. We've maintained yield except this last season we actually had some issues with the wheat, and we're still getting to the bottom of what happened with the wheat. I think I've just pulled some things out too fast. Right, so you've got to watch, you're sort of swinging from one branch to the other, that you're making sure you support the plant and not have that happen. Their organic matter in 2 years has gone from 8.5 to 10.5. It's 20,000 pounds of carbon. Where's it come from? Yeah, from the atmosphere, right? It's free stuff, right? Sugar's being pumped out those roots by microbiology. Right. Some of the sites went down, and it was good that we were measuring it. So this is where they were doing early spring grazing and trampling it with cows, and they did so much damage. The crop barely germinated. This is not a good way to make friends with the cropping boss when you're the cow boss. Right? Cow boss got in a lot of trouble. I'll just go back. So this video is, so they had 1.5 inches of rain before I arrived in early July. The crops were not looking so flash. They got 5 inches of rainfall in 26 hours, and as we drove their property's about 20 miles long. As we're driving, all the neighbors' fields and ditches are full of water, and all their ditches were dry. It was very interesting. So we went and dug some holes and had a look at what was happening at the neighbors. So this is the neighbors' fields. I don't know if you can see. Very bad video. Water everywhere. Mess. All right, so we walked 20 feet to their fields.
Boy, oh boy, oh boy.
I can't believe this. This is a happy now.
Happy now. Happy now.
Let's see. Look how Wayne.
You know how some people have a glass half empty, glass half full. This guy doesn't have a glass. Right, so I love him. He's fabulous, but he's always we're looking for what's not working, and to see those fields absorb water, to have that 5 inches, and they ended up having 11 inches in 2 weeks, and having fields that they had to change their tires, admittedly, but the neighbors could not get on the fields and harvest, and they were actually out there harvesting. Right, and when we dug holes, that top 2 inches is just fluffed up with biology. Right, we can get on our fields. So this is a change in profitability. So their pesticide use dropped by 95%, herbicides 40%, fuel by 20%, and their fertilizer inputs, that's the cost that we dropped their actual input by $100 an acre, and then we put in about $50 worth of biologicals, all the rest of it. And that is only as we transition. So those costs are going to improve. So I have to show this because it's so exciting. All right, so Ian and Di Haggerty, I'm actually going to bring them over in August. These guys are they've got 44,000 acres, Western Australia. Now you're probably listening and you're going, oh, well, you're giving me examples from Canada or New Zealand or Australia. It's not the same here. Everybody says that, right? It is the same. We just have to work with our different limiting things, right? Have you got more issues with mindset here? No, I don't know, maybe. Could happen. All right, so their program, they're grazing sheep. They're putting down the drill worm extracts. So worm liquids at 0.5 gallon an acre. That's it. No trace elements, no other minerals going into the program, and they grow equivalent yields in an average year. In a poor year, they're blowing everyone out of the water, right? In really poor degraded soils like this, like we couldn't even get the bucket to dig the hole. They're putting in 12 gallons of a compost extract as a foliar. Okay, so that's their entire program on 44,000 acres. Now this season, or last season, something very interesting happened. So in summer, nothing grows. So what you're looking at is a summertime photograph. So normally what you'd be looking at would be red sand. This is where the oat crop has come off and they've cut it and taken it away. They got a little bit of summer rainfall. Now in the first year they grew kerosene grass, button grass and windmill grasses. Windmill grasses are also known as crack grass because they climb up your pants and come out your... Australians are great with words, just so beautiful. All right, and as you can imagine, kerosene grass, not something you want in Australia so much. But they let it grow. They just let that land have ground cover. This land had been taking 5 herbicide applications a year, and they could not control radish and they could not control rye. Okay, so massive seed bank of herbicide-resistant weeds. What you're looking at here is where these extracts have gone is a native grass called Serratia, which has not grown in this area for 60 years, appeared and looked like they'd sown it. It had looked like it had been sown, and this was for thousands of acres we drove around and had a look. It's the biological signal for what's going to grow. In this case, a very beneficial grass that they can then feed their sheep on. What grows above ground is a signal that you're giving through your management, biology, minerals, right? I don't know what you people think, but that is the most exciting thing I've seen in my whole life, all right? I just, yay. All right, their stubble doesn't stay around. They have real issues with it, like the neighbors can leave sheep out for weeks and they won't eat the stubble. With these guys, they can only bring sheep in for like half a day because they will take that down to the ground, right? It's nutrient-dense stubble. It breaks down very quickly. So as you know, everything is skyrocketing. Herbicide resistance, pesticide resistance, antimicrobial. Do we need a bigger hammer? Oh, come on, you want to put dicamba and 2,4-D and I'm going to engineer it and that's going to solve all your problems. No, we don't need a bigger hammer. So what they did is they sent away soil that contained seeds from where they hadn't been doing the treatment, so just handfuls of soil with the native seed bank in it, and this soil that had been under their treatment. This is radish, okay? So it's herbicide-resistant radish that's taking 4 or 5 sprays to control. So this is in the untreated area. They're having to pour the herbicide on to control it. Where they've been doing their treatments for 2 years, talking about half a gallon of worm extract, they're killing those herbicide-resistant weeds at half the rate. How cool is that? Right, and we've talked to scientists in the area and no one's coming to look. We're like, you're worried about herbicide resistance? We think we've cracked it. Okay, the microbiology will actually get in there and chew away on those weed seeds. It's food in the right environment. Okay? So we've got to figure out what signal are you sending? How do we optimize plant photosynthesis? We've got to make sure we've got year-round ground cover as much as we can. Root mass, diversity above and below ground, and addressing those limiting factors, right? And what we're seeing across the world, so I'm finishing up, is that markets are getting really, really interested in this. So this is a Western Sustainability Exchange. We got together in Montana with this rancher here, Roger Indraland. Those are the crazy mountains. Patagonia was there. Disney was there. Timberlands, Allbirds, not Ben and Jerry's went at this, but you're seeing those companies ask, how do I get regenerative food and fiber? Where is it? And this question is being asked all across the world right now. These guys are getting paid on carbon sequestration from a project run by Native Energy. Right? They're getting paid for infrastructure development, water, fencing, and being paid up front for potential carbon sequestration. So in Australia recently, Ilana McTiernan, the Minister for Agriculture, came and opened one of my workshops, and this is what she said: regenerative agriculture is the way of the future. Indeed, without it, there is no future for Western Australia. And she gave Western Australia 5 to 10 years before it's down the loo, like they say. Right? And what we're seeing is huge interest in soil health and regenerative agriculture in Australia. Buy my book. It's awesome, by the way. All right. So yeah, thank you so much. Thank you so much for inviting me. I really appreciate it.